Hello everyone, this is Dr. Mayusa Sir, Assistant Professor, MGM's College of Computer Science and IT Nanded. Dear friends, today we are going to learn about a cell division. Cell division is a process by which cell splits into two or more new cells called as a daughter cells. Cell division usually occurs as a part of larger cell cycle. It happens when a parental cell divides into two or more cells called as a daughter cells, which occurs usually as a part of larger cell cycles. In other words, such cycles of growth and division allow a single cell to form a structure consisting of millions of cells. Reasons for the cell division: it's a fundamental process for life that occurs for the growth, repair and replacement in the body, asexual reproduction. So before going to actual cell division process, you have to know about some vocabulary related to the cell division. Somatic cells, these are the cells whose genes will not be passed on a future generation. Germ cells, a cell that is designed to become a gametes, that is eggs or sperms, or a cell whose genes can be passed to future generation. Diploid cells, which is also designated by 2N, a cell with two chromosome sets in each of its cell. All somatic cells in the body are considered as a diploid cells. Haploid cells, a cell with one chromosome set in each of its cell. All gametic cells in body are considered as a haploid cells. So types of cell division. Cell division are broadly categorized into two types, that is mitosis and meiosis. Mitosis, in short, nothing but the process which produce two cells that are genetically identical to the parental cells. Whereas meiosis, it's a process of production of haploid gametes from a diploid parental cell. So gametes are genetically different from parents and each other. So cell cycle, there are two primary phases in the cell cycle: interphase and M phase. Interphase, this phase was thought to represent the resting stage between subsequent cell divisions. But the new research has shown that it is a very active phase. And the second phase is M phase, that is also called as a mitosis phase. This is where the actual cell division occurs. There are two key steps in this phase, namely cytokinesis and karyokinesis. So mitosis is a phase of a cell cycle where a nucleus of cell is divided into two nuclei with an equal amount of genetic material in both the daughter nuclei. It succeeds the G2 phase and is succeeded by the cytoplasmic division after the separation of nucleus. Mitosis is essentially for the growth of cells and the replacement of worn-out cells. Abnormalities during mitosis may alter the DNA and resulting in a genetic disorders. So what are the features of this mitosis? In each cycle of cell division, two daughter cells are formed from the parent cell. The cell is also known as equational cell division because the chromosome numbers in the parent cell and daughter cells is the same. In plants, mitosis leads to the growth of vegetative parts of the plant like root tips, stem tip, etc. Segregation and combination do not occur in this process. So now we will go towards each and every step of mitosis in detail. The different stages of mitosis occurring during the cell division are prophase, metaphase, anaphase and telophase. So first one is a prophase. Prophase immediately follows the S and G2 phase of a cell cycle and is marked by the condensation of the genetic material to form a compact mitotic chromosome composed of two chromatids attached at the centromere. The completion of prophase is characterized by the initiation of the assembly of mitotic spindle, the microtubules, and the proteinaceous component of the cytoplasm that help in the process. The end of prophase is a breakdown of a nuclear envelope. The second step is a metaphase, but before that some small changes occurs in the chromosome in the prometaphase. In the prometaphase, the nuclear envelope disintegrates. Now the microtubules are allowed to extend from the centromere to the chromosome. The microtubules attach to the kinetochores which allow the cell to move the chromosomes around. Next one is a metaphase. At this stage, the microtubule start pulling the chromosomes with equal force and the chromosome ends up in the middle of the cell. This region is known as the metaphase plate. Thus, each cell gets an entire functioning genome. Next is anaphase. This is the third phase of mitosis. The splitting of sister chromatids marks the onset of anaphase. These sister chromatids become the chromosome of a daughter nuclei. The chromosomes are pulled towards pole by the fibers attached to the kinetochores of each chromosome. The centromere of each chromosome leads at the edge while the arms tails behind it. Last, telophase. The chromosomes that cluster at two poles start coalescing into an undifferentiated mass as the nuclear envelope starts forming around it. The nucleus, Golgi bodies and endoplasmic reticulum complex which had disappeared after prophase start to reappear. So these are some phases of mitosis. Now we'll go towards the function of mitosis. There are two important functions of mitosis. First, mitosis helps in the development of an organism. In single cell organism, mitosis is the process of asexual reproduction. Second important function is mitosis help in the replacement of damaged tissue. The cells near the damaged cells begin mitosis when they do not sense the neighboring cells. The dividing cells reach each other and cover the damaged cells. Significance of mitosis. Mitosis is responsible for the development of zygote into an adult. Equal distribution of chromosome to each daughter cell. Next significance, it is responsible for the growth and development of an individual. It maintains the constant number of chromosomes in all body cells of an organism. Mitosis is required for asexual reproduction, vegetative propagation in plants, and is also responsible for the repair and regeneration of damaged tissues. Mitosis helps in maintaining the purity of a genome as no recombination or crossing over takes place. It is responsible for the repair and regeneration of old and damaged cells in the animals. Example, gut epithelium, blood cells, etc.